Good morning, everyone. Bonjour à tous. Mon nom est Yukiko Sato. Je suis doctorante dans le laboratoire de David Thomas et John Hanrahan, ici même au complexe des sciences de la vie.
And my name is Brendan Cordero. I'm a fourth-year PhD student in Professor Samantha Gruenheid's lab here at the Life Science Complex in the Complex Traits Group. Welcome to McGill University's Life Science Complex's 10th Anniversary Symposium. Nous sommes très honorés d'animer le Symposium de ce matin. Today, we celebrate the original vision and exceptional support that brought the state-of-the-art complex to life. And we will now pull back the curtain to showcase some of the fascinating research that is going on inside. First, however, we have the great pleasure of welcoming McGill's principal and vice chancellor, Professor Suzanne Fortier, to the podium to officially open up today's events.
Bonjour à vous tous, good morning, buongiorno a tutti. C'est un grand plaisir de vous avoir ici aujourd'hui pour cette belle célébration, 10e anniversaire du complexe des sciences de la vie. What a thrill to welcome all of you here, not only this morning, for the whole day, because, of course, the best way to celebrate this anniversary is to showcase what has happened here and what is yet to happen here, thanks to all of the talents that we can bring together in the life sciences complex. Let me acknowledge first our very special guests who are here with us this morning. Francesco Bellini, Dr. Bellini, who is here with his wife Marisa, as well as his sons Roberto and Carlo. Bienvenue, benvenuti. Also with us is Morris Goldman, who is here. As you know, Morris has played a big part in the cancer center, the Goodman Cancer Center, and he's here with his son, David, and with his daughter-in-law, Mia. Bienvenue, welcome. So it seems like yesterday, but in fact it was 10 years ago, in 2008, that the community assembled here to celebrate the opening of this new center, the Complexe des sciences de la vie. C'était un des plus grands projets de construction de l'histoire de McGill. So it was really quite something to celebrate for us. And the vision behind this complex is really inspiring. It was to bring under one roof all these talents from different areas, from the medical area, from the basic science, from the life sciences, so that these people could work together and really accomplish the vision of bringing the discoveries, the knowledge, all of the big questions that we're addressing here, bring those to society and make sure that first and foremost, we're all thinking about the patients out there who really count on us to make progress in our knowledge of the health sciences so that we can bring solutions to the many problems we have. And we will hear throughout the day all of the researchers presenting their work. We all know, of course, and about all of still the many challenges we have, the devastating illnesses that we still have difficulty in confronting. And so it is extraordinarily important that we bring talents together, all of the effort, the hard work of these people to address these issues. And we're so proud that it's happening here in Montreal, here at McGill in this life science complex. In a few minutes, I'll turn to you, Francesco, but let me also say that we'd like to share our appreciation to the many people who've participated in the success of this and in the creation of the life sciences complex. The government of Quebec, Canada Foundation for Innovation, Anthony and Deborah Campbell, La Fondation Jean-Louis Lévesque, and the Dr. John R. and Clara M. Fraser Memorial Trust, the estate of Phyllis Butterworth, the estate of E. May Covert-Duc, and the estate of Marianne Fortin-Jouer, and the Townsend Lamar Foundation. I want to say a big thank you to these people to have contributed to this creation of this center. You know, in order to have so many people who want to participate, you need a catalyst, you need a person who can mobilize the support, And I'd like to acknowledge Francesco Bellini, Dr. Bellini, as the person who played that role with a very generous gift of $10 million. This was in 2002, and this was the catalyst to bring this life science complex to life. People then join in under your vision, under your influence, under your leadership, join in to help us accomplish that. Dr. Bellini is a respected scientist and the founder of several drug companies, including Biochem Pharma. We all know how his company revolutionized the treatment of AIDS with its drug 3TC. You know, last night we were talking about 3TC and the fact that, at least for myself, this was the first time that I heard about this approach of using a cocktail of drugs. And I was curious to know if this was started with 3TC and the treatment of AIDS, and indeed it was. So not only was there a major discovery, a major contribution here to the treatment of AIDS, but a new approach in bringing other drugs together. And today, I'm hearing about this in other areas, but this was a first here, thanks to Dr. Bellini. Now, the family not only contribute a very generous sum of money to help us make this possible, but also a vision. And you had a particular vision on how this place should be designed. Not only the brick and mortar. The brick and mortar was only a consequence of your vision, a vision that was to have a complex that would be very open, so that people would be working together. Increasingly at McGill, we call that the creative collisions that we need to see happening among people who might not otherwise meet each other, but they meet each other, and then all sorts of new ideas, new way of doing things are launched. And this is the vision that you had from the beginning. And this vision is one that benefits, I would say, first and foremost, all of our students, postdoc, young scientists who are part of this complex and are really working in this kind of environment and are making discovery in this kind of environment. Now I'd like to leave the podium to Dr. Francesco Bellini so that he can once again be a source of inspiration. Grazie.
Thank you, Susanne. Thank you, Susanne. You know, for me, it's a kind of very proud to be here again after 10 years. You know, it is, I'm very happy and I'm very sad. I'm very happy for what, you know, has been accomplished. This is a beautiful building. It's very successful. I'm sad that I am 10 years older. You know, some of the faces which they were there, they are still here today, and they all look good, okay? I've been told 10 of your success, but I wanted to tell you that not very many people knows that the biggest, I think, AIDS drug was discovered here at McGill, okay? And was discovered in the old chemistry, and then we took it over and we developed. We tried to make it a joint venture together, and it did not work out. But 3TC became a major product, a major drug. And you know, center like this should be, you know, the places where those type of discovery should be made. Me, I think the university should do the research, should be a very good research place. And then ideas and the discovery should be developed by the private sector. But the university should have a slice of it. in form of royalty, in form of whatever it is. In that way, the circle is closed. It's an initial investment, which is made in R&D, okay, not in R, and that cycle can continue, and it's going to be financed by the product which eventually goes to the market. And was unfortunately then Miguel did not get much out of it, and that's why I came in personally, and I made this contribution for this building. And here it's nice to see that new people came here. I've been told, because these are real state-of-the-art facility, and young people from all over North America, Europe, and the world. They are working here. They are doing research here, and they are bringing science ahead. And as I mentioned to you, we should have much, much more, except we live in a nation which does not spend very much for research and development. And the future is there. That is the future. We want it or we don't want it. The future is a new product, new technology. We have disease which, antibiotics in a few years, we are obsolete. Okay, we need a new thing to come out. We need a new anti-cancer agent. It's plenty, and we don't, as a nation, we don't spend very much. And I hope you are the guide, and whatever is happening here is going to happen other place too. To real have this place, many people does not know. I was living in Quebec, and I left Quebec to go in Alberta, and one of these because I had a huge argument with the Charest, and he did not get it, what it is a province, was. Quebec was one of the leaders in biotech in North America, and we just lost it, most of it. But I'm back, and I started again, 10 years older, whatever it is, but I have 2 sons which they are taking over, and I hope that they can do beautiful things for this country, for this province, city, this country. I thank you very much for coming all here this morning, and enjoy today, and thank you.
Thank you very much, Dr. Bellini. Nous souhaitons maintenant inviter Dr. Adelman, vice-principal santé et affaires médicales et doyen de la faculté de médecine, à dire quelques mots.
Merci beaucoup, Yukiko et Brendan. Au nom de la faculté de médecine et de l'Université McGill, c'est un grand honneur pour moi de célébrer avec vous le 10e anniversaire de ce complexe de sciences de la vie. Et on est en compagnie de personnes très importantes. First of all, we have the Goodman family are here, and the principal already mentioned Morris and David and Mia, who are here. But I also want to give a thought to Rosalind, who isn't here, who really played such a key role in the conceptualizing of the Goodman Cancer Center and really being a presence here while she was alive, almost on a daily basis, and really an important part of this environment. Of course, also want to acknowledge the Bellini family, Marissa, your sons. I think your words just now were exactly correct. We have to find a way of making this province understand what it is to be a 21st century society which invests in the future. I also want to acknowledge our government partners and guests who have supported us, and of course the outstanding scientists, the students, fellows, staff who are here, who really are the people that make this place what it is. So the idea behind this Life Sciences Center was to create a world-class hub that would attract top researchers. I noticed that a lot of the pictures, some of the researchers are still here, they look a little bit older, but there are many, many new young people here. There's 60 principal investigators that work in the Bellini and Goodman Cancer buildings, and 500 staff, 500 leading scientists at various levels. And this, together with the 2,000 people who are in the adjacent McIntyre and Strathcona buildings, really creates an extraordinary ecosystem, a critical mass of world-class research that gives us an opportunity here in Montreal, here at McGill, and in Montreal in general, to really make a difference and to build on the vision that Dr. Bellini was just talking about. I would just remind those who are not, I see many people who work here, so you'll indulge me for the people who don't, remind people that there are 5 major themes in the Bellini and Life Sciences Center. So first of all, there's cancer research, and the theme within the life sciences is led by Michelle Tremblay, but of course Mariette Park is here, and she's the actual overseer and boss of the Goodman Cancer Research Center itself. We have the cell information systems led by Alvin Schreier, chemical and structural biology, which is now led by Albert Berghaus, complex traits, which is led by Sylvia Vidal, and developmental biology, which is led by Paul Lasco. When you really look over the last 10 years, there have been a lot of scientific accomplishments. So there's novel approaches to Fragile X and autism, Parkinson's disease, cystic fibrosis, schizophrenia, Alzheimer's, inflammatory bowel disease, influenza, respiratory disease and malaria, epilepsy and aging. So a very broad scope of activity in which basic science has really been harnessed to address major and important problems that face our society. And those who are going to be lucky enough to stick around for the presentations later today will really get a taste for the excellence that we have here at the Life Sciences Complex. Of course, the Life Sciences Complex project would not have seen the day without leadership. We've already heard about the leadership of the university and the leadership of Dr. Bellini, the Goodmans, and so on. But there's also the scientists, and we're going to hear later from David Thomas, who was the original PI lead for the chemical and structural biology theme. And he, together with Professors Tremblay, Lasco, Schreier, Philippe Gros, who's here, who was the original lead of the complex trait theme, they were the ones who really pulled this thing together and got the CFI and so on. They also had tremendous support from former Dean of Science, Alan Shaver. You saw his picture was up earlier, and also Abe Fuchs, the former Dean of Medicine. And their successors, Dean Levin from Medicine and Dean Grant from Science, who were here at the launch. Alors, les activités du complexe sont le fruit d'un énorme effort collectif, non seulement du corps professoral, mais aussi des membres remarquables de notre personnel, dont la plupart sont ici aujourd'hui. This was and continues to be a team effort, one that really requires the involvement of people from every different level within the Life Sciences Complex to make it a success, and it is a great success. Alors, félicitations à tous. Congratulations on this wonderful 10th anniversary. And thank you very much. I'm now going to introduce my colleague, the Tomlinson Professor of Chemistry, Dean Bruce Lennox. Bruce.
Thank you, David. Principal Forte, special guests, Bellini family, Goodman family. It's a great pleasure for me to be able to speak for a moment about the place and the role of the Faculty of Science in this fantastic life sciences complex. Joining me today, you can see in the back are a number of students, staff, faculty associated with the Faculty of Science. But the one principal group that has occupied space in this complex has been the developmental biology group headed by Paul Lasco. This group has really pioneered a number of approaches within developmental biology as it interfaces to life sciences and health sciences. What they had set out to do is to understand how genes in the inner machinery of our cells, how they influence every stage of life, from the developing embryo all the way to the aging process. So by understanding how and why things go wrong with these processes, there clearly are entry points, very, very important entry points into the first steps of prevention and perhaps treatment. The LSC has been pivotal in building a capability that's truly world-class, and you can only do that if you have all the ingredients of world-class research. That starts with our researchers, our principal investigators, our professors. One thing that when we're out in the world searching for talent, we have many components. We have the reputation of our institution. We're competing with the best in the world. We have the reputation of our students. We're competing with the best in the world very, very well. But we also have an ingredient that's not publicly discussed that often. That's the facility. 10 years ago, 12 years ago, 15 years ago, when the vision of this complex was put forward by Dr. Bellini and my colleague Alan Shaver and Dr. Fuchs, we weren't just competing for the talent and the students, but we were competing against dozens of world-class buildings. That has changed. We have, just within the Faculty of Science, we have recruited some incredible talent who now are working in this building. So the expression of leveling the playing field, I think we've tilted it. I think you've tilted it in our favor. And as we'll see throughout the day when we hear presentations, especially from the developmental biology group and the posters, you can see that we're doing incredibly well because of this tilt. So thank you. A couple of snapshots, and you'll see many more throughout the day, of really the great successes of the developmental biology group. Gary Bruhard in his lab uncovered the role played by a genetic mutation in causing a microtubule defect. This in turn revealed a very rare form of epilepsy. So this has the potential of providing an entry into therapeutics for diseases like this and others. And Siegfried Hikemi, my colleague in the Department of Biology, has been studying the metabolic basis of aging at the cellular level. In this work, Siegfried and his research group have obtained really compelling evidence that the textbook version of aging is being associated directly with free radical chemistry, with oxygen-free radical chemistry. This is actually, he's shown very, very clearly that this is not the case. And in fact, many of these species can be used to combat aging. So this is a head-turning moment in science. There'll be many, many approaches now in terms of understanding aging that will actually lead to, I think, entirely new streams of therapeutics. These are just two of the many, many snapshots that the LSC has enabled in the last 10 years in terms of really groundbreaking research. So on behalf of the Faculty of Science, Dr. Goodman, Dr. Bellini, and your families, I want to thank you. Your contributions will provide an incredible legacy that you'll see today, but a legacy that I think will be decades long. So I'd now like to introduce Dr. David Thomas. He's Canada Research Chair in Molecular Genetics, and David was one of the driving forces behind the successful design and the actual building of the Life Sciences Complex. David.
Thank you for that introduction. Chers invités distingués, chers collègues, et bonjour à tous. So there's a history of the development of the Life Sciences building that exists. Maybe you don't know about it a little bit before. So I moved to Montreal in 1984, and as part of the National Research Council's initiative to build a biotechnology institute here. And in fact, the site we chose was here, these buildings along here, and we looked into it and ended up actually renovating a wing of the Royal Victoria Hospital when we found out that these are actually heritage buildings out here, and we could not knock them down. So, but in our original CFI application, we used the footprint that we'd looked at for the NRC. But of course, then we had to change, and we changed to this site over the garage. There's a history to that, a family history to that too, but I won't go into it right now. So the idea was, I think it was Philippe and Michel, even before I arrived here, were very keen to get a new building going. And bringing in Paul Lasco and a number of other colleagues, sorry, I'm going to apologize right up front. There were a huge number of people that helped us at various stages. And if I could have the first slide, do I just do that? I don't do that. Yeah, you probably can't see this too well. But here we were 10 years ago, looking a lot better than we do now, I guess. Some of us, not all. And this was really, as I was trying to emphasize, is a team effort, and I certainly can't, both on the side of the scientists and the side of the administration at McGill, there were certain roadblocks that we encountered, but being stubborn and persistent, we got things rolling. We hired some really good people, a manager, Carmen Lampron, who would see through the more complicated stages. The architects provided, the engineering firm, Pomerleau, provided us with Jean Ouellet, who I think may be here, and he now works at McGill. And he was really a driving force in getting things done. So we ended up with a building that was built after we started on time and actually on budget, which I think Suzanne Forte referred to as probably unique in the history of McGill. And so this was not without some sort of disputes between us, but it worked out very well. But the original vision was really, as you've been told several times, was a multidisciplinary building where scientists, student scientists, of course, would interact and be productive and give a boost to our hiring of exceptional people. Not that McGill in the past has not hired exceptional people. And to provide a kind of a forum where people would really come and interact at different levels, right through what I'm looking at right now. I actually didn't move into the building. I had more space over in the McIntyre, so I stayed there. Sorry. So just to go on to the next, whoops, why doesn't this work? Oh, yeah. So the purpose, as Dean Adelman said, was 5 multidisciplinary research themes. And why do we choose these and not take conventional departments? It was because we felt that the future lay in some sort of degree of multidisciplinarity. As you are all aware, I think at McGill, people tend to be kind of independent, and and sometimes you can't sort of expect them to lead or to follow some sort of leadership, but what you expect is that a high degree of collegiality, which we do have, of course, which is one of the distinguishing features of McGill. We also would have well-managed core facilities, and that is that we would hire, and you will see if you take advantage of the tour, we will see, for example, our imaging facility, which is second to none. I was just at the Crick Institute, the new Crick Institute in London, which combines one of the institutes where I worked in London, And they have adopted some of our characteristics of what we planned here. It's a much bigger building. There's 1,500 scientists working there. It's a huge building, in fact. And for the translational aspects, they actually hired an old colleague of mine that was working at GSK, who is running a translational part to their scientific themes, which are similar to our own. And I should just sort of mention there, one of our really kind of completing things in the structural biology and chemical biology theme has been to bring in a chemist, a well-renowned medicinal chemist, Bob Zamboni, who was the VP research at Merck Frost. And this has been, I think, transformative in really opening our eyes as to the advantages of having a translational, as at the CRIC, having a translational aspect to our research. Okay, what have we delivered? Well, the building, you're here in it. Modernized laboratories through other CFIs and other programs, governmental programs. So we've renovated the McIntyre and Stewart buildings. We have a life sciences complex, which works well, the translational research facility, the generosity of the Goodman family to the Goodman Cancer Research Center. I think I've misspelled something there. And we have a whole roster of national and international collaborations where McGill plays a very important leadership role. And of course, spin-off companies, which I think Dr. Bellini was kind of guiding us to thinking about where the fruits of our research are going to be exploited. Obviously, this is going to be done by the private sector, but maybe we have to move the bar a little bit closer to the real applications of our research than we have up till now. Some of us, others, of course, are taking advantage. So here's our generous donor, and here's the molecule that Bernard Belot, I believe, a graduate of biochemistry but a professor in chemistry, developed, and was then further developed by Dr. Bellini. And Maurice Goodman, I believe this is ribavirin, and this was developed as well. You know, we've talked a lot, and I'm sure about your vision and whatever. I would like to invite you, actually, to deliver on the vision. Come and teach a master class to some of our graduate students about what it takes to be an entrepreneur. Yeah, I think we really need real-life examples, and I think an hour or two spent in an afternoon with some students would be really very, very useful. Sorry, this is off. Yeah, so what next? You know, as that great Canadian philosopher Wayne Gretzky said, you know, don't skate to where the puck is, go to where the puck is going to be. So what are we going to do next? So this afternoon, you'll hear from Michel Tremblay. You'll hear about our initiative in stem cells. We have a structural biology center right up here being run by Martin Schming. Cystic Fibrosis Translational Research Center, of which I'm a member, a declaration of conflict of interest maybe, but which is directed by my colleague John Hanrahan in physiology. A combined initiative in orphan diseases that we're talking about with people in biology, with Paul Lasco. And a number of, as I've said several times, the translational research projects are really important, I think, in moving things, as I said, a little bit more to the applications. And Morag Park, the inimitable Morag Park, is putting together, I think, all the cancer, trying to put together all the cancer institutes in Montreal in some sort of consortium. I'm sure she'll succeed. And that's a repetitive thing. So here we are. Here's a beautiful day. I think sun is just rising. I don't see any lights on right now in the labs, but I'm hoping people are going to start. And just to thank again our donors and the participants and all the people that have really made this happen. And I think you can all applaud yourselves. Thank you.
Thank you, Professor Thomas. And now for a look inside of the science being conducted in the magnificent space, we would like to introduce you to today's speakers, each representing one of the complex's 5 themes. First, we'll have Dr. Arjun Krishnaswamy, Assistant Professor of Physiology and Tier 2 Canada Research Chair in Neural Circuit Assembly and Function. Recruited from Harvard University, Dr. Krishnaswamy represents the Cell Information System theme. Next, we'll have Dr. Ilana Want, Associate Professor in Biology, who received her PhD in Neuroscience from Brandeis University in the USA and pursued postdoc work at University College London. Dr. Watt represents the Developmental Biology theme. And then we will have Dr. Ian Watson at the Goodman Cancer Research Center, Assistant Professor of Biochemistry and Canada Research Chair 2 in Functional Genomics of Melanoma. He completed postdoc work at MD Anderson and Dana-Farber. Dr. Watson represents the Cancer Research theme. We'll then have Dr. Albe Guarne, full professor in biochemistry, who came to us from McMaster's and completed her education and training in Barcelona, Spain, and the United States. Dr. Guarne represents the chemical and structural biology theme. Then lastly, we'll have Dr. Jörg Fritz, assistant professor in the Department of Immunology and Microbiology, who came to us from the University of Toronto and prior to that, l'Institut Pasteur, Everis in Vienna. Dr. Fritz represents the complex traits theme. Les 5 responsables présenteront maintenant leur thème de recherche, à commencer par le professeur Arjun Krishnaswamy.
Okay, thanks for that introduction. Thank you to the organizers and the university brass for putting this together and extending the invite. Also thank you to our donors who have played such a critical role in setting up this life sciences complex. As you heard, I'm probably one of its newest members. I was recruited out of a fellowship at Harvard about a year and change ago. And in short, this is an excellent place for a young scientist to set up shop. The modern facilities, the access to advanced core equipment for microscopy and genome modification, as well as the interdisciplinary mix of excellent colleagues, both on the ground floor where I'm located and all throughout the building, make this an exciting and stimulating place in which to conduct our research and forge new collaborations and new ideas. So I'm a member of the Cell Information Systems Group, and broadly speaking, we're interested in the way that biological systems encode, transmit, and act upon information at the cellular and organismal scales. We're curious about how cells develop these structures and how they might go wrong, as happens during disease. Our contribution to the theme is to study it in the context of the brain. We'd like to know how brain cells, called neurons, form the systems over which they exchange information, called circuits. And we'd like to know how these circuit-like patterns arise and how they function as they must in order for our mental abilities. Things that span from as simple as reflexes through sensation and motor control, perhaps all the way to more cognitive kinds of things. Now, the connections themselves are set down early in development and revolve around the choices that neurons make, choices about who they should connect with. But the factors that lead to those decisions, the coordination needed to bring many neurons together, and indeed the consequences of some of these early brain wiring events for mature function, and as happens all too often, dysfunction, are truly major open issues and are the issues that really drive our work. So we try to study these problems by using the visual system of mice, and there's all kinds of reasons for that, but basically it makes the experiments easy, and you'll see that in a moment. In vision, it begins in a thin sheet of neural tissue located on the back of the eye. It's called the retina. Images of the world are focused there, and if I were to take a cross-section through it and cartoonify it for you, you'd see that all the neurons are grouped into 3 layers, an outer layer containing photoreceptors, an inner one containing interneurons, and a lower layer containing retinal ganglion cells whose axons bundle together to form the optic nerve and send visual info off to the brain. Now, we actually know a great deal about the organ. We know a lot about its circuits, too. All of them begin with a photoreceptor. These neurons are photon detectors. They're like the pixels in your iPhone camera. Every single one of these circuits ends with a retinal ganglion cell, which, unlike the photoreceptors, come in a variety of flavors or cell types. And strangely, each of these types appears specialized to detect just one feature in the visual scene. This could be edges with a certain orientation, objects that move in a particular direction, colors, patterns, contrasts, so on and so forth. Now, they detect these features completely independent of one another, Okay? The pink cell doesn't know the other ones exist, but are arranged in parallel. And what this means is if we were to label the pink guy across the retinal surface, we'd see, if we could see it clearly, that the cells arrange themselves all across the retina. The thought is that he's trying to detect his feature from all points in visual space before sending that off to the brain for further analysis. Now, all the types are doing this, and what it means is that the eye isn't a camera. It doesn't take a picture of the world. Instead, the eye is a sophisticated neural computer. It decomposes a painting like this into approximately 30 representations, with each of the images in this deck containing emphasis on a particular aspect of the painting, which remarkably reflects the feature preference of one of these little neurons here. And so we might imagine there's a cell in there that likes the low spatial frequencies. It sends a blurry version of the Lisa upstairs to the brain. There's one that likes the green images, the green colors in the image. If the Mona Lisa were moving around, there'd be one that would encode the moving edges in the image, and so on and so forth. Now, what the brain does with information like this, how it flattens it back and gives us this percept, or whatever it is a mouse sees, is something I'll touch on at the end. Much of the immediate term work in the lab, and in fact the next couple of slides, are going to be about the first mystery, which is how on earth these neurons even acquire these remarkable abilities. To make a long story short, they're endowed with their preference. They're endowed with their preference for visual features because of connections they form, circuits they make, with a very specific mixture of interneuron types. The basic question in our lab is to understand which types wire into a given ganglion cell. What are the genes that direct that kind of process? And given that a cell like this wires selectively to one like that, what is his contribution to this cell's feature selectivity? These are the kinds of things we want to learn more about in the next couple of years. But in the next couple of slides, I'll expose to you just one method that we use to gain insight. So if we wanted to know which ones of these connect with those, we would need a way to map out circuitry. And without going into detail, it turns out that a revolution in genomics allows us to color mouse retinae, just like I've colorized this cartoon over here. And so in the next slide, we're going to examine this pair in which I've labeled both neurons with green fluorescent protein, and here they are. Here's the ganglion cell, here's the interneuron. With tools like this in hand, we can label this neuron with green, and then inside this cell, install a light-sensitive ion channel, which allows us to control neural activity in this neuron with light itself. Combining this genetic tool with next-generation two-photon microscopes that we build in-house. Come by the lab, I'll show you how they work. Combining it with these allows us to record from a single neuron and stimulate hundreds or thousands of these individual neurons over here. So here are a bunch of dots, we're stimulating a bunch of neurons, and we can map out the connectivity to the ganglion cell. Strangely, not all of these connect. In fact, only a small subsector of them do, and they connect with a very pronounced tilt. And without going into detail, it turns out this tilt is absolutely critical for giving this ganglion cell a sensitivity to objects that move like this. Now, wiring patterns like this allow us to then return to these neurons and analyze the genes that they express. We're looking for wiring genes. And without going into detail, we've discovered a number of what are called recognition molecules that allow neurons like this to find and wire selectively to neurons like that in patterns like this. And again, without the detail, it turns out that many of these molecules are commonly mutated in forms of neurodevelopmental disorders such as autism. And I'm happy to tell you more about that afterwards if you're curious. What I'd like to do in my last slide is to just give you a sense of where we're going. If you invite me back 10 years from now, I can tell you more. The success we've had in moving between circuit patterns, circuit function, okay, and wiring genes makes me very curious about what the brain does with a signal like this. How does it flatten it back into the image? How does it combine it with internal states such as attention or anxiety or motivation? These are the kinds of things I want to tackle next. And to do it, we're building new kinds of microscopes that allow us to peer into the brains of mice, where we've labeled the neurons with a dye that lights up when the neurons become active. Doing so inside mice that run through virtual reality hallways as they experience a visual environment, and reading from the neural code will allow us to come up with a mechanistic understanding of how signals like this get transformed and guide behaviors like that, providing a good framework for understanding what might go wrong in cases of illness. So that's the future, but for the present, I'm going to stop and thank my former advisors, my lab, collaborators, essential core facilities here at the LSE, and some funding. Thanks very much.
Do we have any questions for Dr. Krishnaswamy? Perfect. Thank you very much.
Hello. It's great to be here. And so my name is Ilana Want, and I'm an associate professor in biology. I was recruited to McGill about 7 years ago and was absolutely thrilled to join the Life Sciences building. My lab is located up on the second floor, just up above in Bellini. And I really want to extend thanks to the donors and in particular to Dr. Bellini for having the vision to create this really amazing research environment. And I was thrilled to join it, and I'm still thrilled to be here because it's a really exciting and dynamic and intellectually rich environment that I really enjoy doing science in. So I thought it would be most fun to tell you a little bit about the science that we do in my lab. And this is actually all unpublished work, quite new work that we're really excited about, and I hope you're going to find it interesting too. I'm happy to be kind of representing my developmental biology colleagues, but this is just a story one of the stories that I've chosen to tell you about. So in my lab, we're interested in the cerebellum, the little brain at the back of the brain that contains about half of the neurons in your brain, and is famously involved in things like motor coordination and motor learning, among other things. So we don't study this in people, we study this in mouse models, and here I'm showing you a picture of the principal cell of the cerebellum, Purkinje cells. And Purkinje cells, you can see they're lined up beautifully, and they have these beautiful axons that I hope you can see. These are the axons that carry the information encoded in the cerebellum out of the cerebellar cortex. And I hope you might be able to see that there are some interesting structures in those axons, these big swellings. And those have been named torpedoes. They're actually named torpedoes about 100 years ago. They sound pretty bad, right? Torpedo. Well, that's because they have been seen in many different human disorders. So if you look at post-mortem tissue from patients with Parkinson's, Alzheimer's, many forms of ataxia, other disorders, you see these axonal swellings. And so this and other things have led people to this kind of idea that these torpedoes are pathophysiological. They contribute to the neurodegeneration that occurs in these diseases. So we came to this project because we also have a really great 2-photon microscope that allows us to look at axons at the micron resolution and look at them in a functional way. So now we're going to have a little introduction to neuroscience, and I'll ask you, what do axons do? Now, if you ask 10 different neuroscientists, they might come up with 10 different answers because axons do a lot of things. But I think we can all agree that one of the most fundamental things that axons do is carry information across space. So we wondered, just to start, what might a torpedo, one of these big swellings, do to that information being carried across space? Computational neuroscientists have modeled this, and they thought it was likely that this caused the axons to misfunction. Part of the neurodegeneration. It makes sense. So we were able to target the axons and the cell bodies and the axons, and actually record that activity moving across space. And we could look at axons that have torpedoes and axons that don't, and just see how that might impact function. So here I'm showing you a control axon. So there's no torpedo, and you can see there's an action potential at the soma, and then very, very soon after, it's there along the axon. We can detect it. But every now and then, you see an axon in the soma that you don't see along the axon, and that's called a failure. So really what it is a failure of information to be transmitted along the axon. That's a problem. It means it's not working perfectly. We don't have a lot of them in Purkinje cells, but we do have some. So what happens when there's a torpedo? So what we were expecting was that we'd see a lot more of those. But that's not at all what we saw. In fact, we had to look really, really hard to find any failures at all. So contrary to what we expected, we actually saw that the failure rate became basically zero, and that torpedoes, this big swelling in the axon, doesn't appear to make the axon work worse. It actually appears to improve axonal function. So that's kind of interesting, and it suggests, well, if the axons are actually sometimes misfiring, and sometimes when that happens, we see a swelling, and sometimes the swellings, we don't see that. Maybe that misfiring actually causes a swelling to occur. So we did some pharmacology to mimic axonal failures. And what we found was that if we look at an axon over time, this is several hours, in normal, without any axonal failures, we don't see any changes. But if we mimic high axonal failures with pharmacology, we actually can create axonal swellings. So this really suggests to us that this is a kind of homeostatic adaptation. The axons are misfunctioning, and when that happens, there's a response that actually is going to improve function. Fine and well, but does this have any actual impact for the circuit when it's functioning? We wondered about that, and so to look at that, we actually did a very simple motor control and motor learning task called Rotarod. And this is what we do. We put mice on a rod, gently turning rod. It takes quite a bit of motor coordination for them to stay on, and you know, some mice are better than others at this. When we do this day after day, some mice, all the mice learn, but some mice learn a lot more than others. So does this have anything to do with torpedoes? Well, we found, in fact, that it did. So we found that the mice that had more torpedoes actually learned more. So we could separate the mice into high learners and low learners and show that those torpedoes were actually associated with improved cerebellar function. So the old working hypothesis that these are a marker of neurodegeneration and a negative thing for circuits, we think is wrong. So our new hypothesis, I hope I've shown you some data to convince you, is that this is actually a positive thing. This is the neuron responding to some misfunction and fighting back, making a torpedo that actually helps it improve cellular function and improve brain function, actually. So, of course, this work was not done. Oh, I'm going to just skip. We're now looking at some new. We have some really fancy new microscopic techniques that are going to allow us to look in even more detail at these structures, not just a couple of axons and a couple of torpedoes, but the whole cerebellum. And all the torpedoes in the cerebellum, all the axons and torpedoes in the cerebellum. And this is with our new light sheet microscope, which is just up on the second floor here in the Bellini building. So finally, of course, I want to thank all the people, who have made this happen. This is my lab. The people in red are the people who've done this work, and most of the work I've shown you was done by this graduate student, Dana Gleng-Ouellette. I also want to, of course, thank my funding sources and collaborators, and also special thanks to the imaging facilities, the CYAN and ABIF here in the Life Science Complex, who've really helped me get this work going. So thank you very much.
Do we have any questions for Dr. Watt? Excellent.
So first, I just want to say it's a great honor and pleasure to present on behalf of the Goodman Cancer Research Center and to discuss a little bit about my research that focuses on melanocyte biology. So melanocytes are the melanin-producing cells in the body. These are the cells that provide tanning in response to UV and sun exposure. But they're also the cell of origin of the most deadly form of skin cancer, which is cutaneous melanoma. So I've been in the cutaneous melanoma field now for 8 years, and it's actually been very inspiring, the amount of progress that has been made translating basic science discoveries to the clinic. And this is best demonstrated by the advances in targeted and immune therapy. So I'm sure many of you have heard in the media the advances in targeted therapy. This is where you understand a patient's genetics and prescribe a drug based on a specific mutation. Immune therapy is a type of therapy where you reactivate a patient's immune system, getting your immune system to control tumor growth as well as killing tumors. And the advances in melanoma has really changed the way we think about diseases. It's been a model disease in terms of cancer, about how we think about treating other types of cancers. And what you're seeing here is a cartoon of the responses to targeted immune therapy seen in clinical trials with patients. And with targeted therapy, what you see is you see an amazing immediate response. Tumors often melt off of patients. Unfortunately, a small subset of patients only have durable long-term responses. On the right, you're seeing the survival outcomes for patients with immunotherapy, which at first may seem less impressive. But what you see is this plateauing effect. That is a fraction, about 20% of patients in initial clinical trials were having long-term durable responses that are actually going out 15 years. And with new combination therapies, these are actually higher, 30 to 50%, having these long-term durable responses, essentially curative therapies. Now, when I started in the melanoma field, the median survival for stage 4 metastatic melanoma was 6 to 9 months. But now, with these new types of therapies, we're starting to think about curing more and more patients. And it's really set the bar in terms of cancer research, where it's no longer appropriate just to develop therapies that have an immediate short-term impact, but we need to develop therapies that get this plateauing, long-term durable responses. And the current goal of the research community is to develop therapies that result in long-term durable responses for the majority of patients. And this is the bar that the melanoma community has set. So how are we doing this? Well, one, we're trying to understand the genetics of melanoma. We all know that cancer is a genetic disease. It's caused by specific mutations in genes, and this is best illustrated by the work that I did when I was a postdoc at MD Anderson Cancer Center through the TCGA. And so the TCGA was a joint NIH-NHGRI sponsored project whose goal was to characterize over 30 cancer types, including melanoma, at the DNA, protein, and mRNA expression. And this was my first experience with regards to big team science. So this involved over 90 institutions, over 300 investigators, where we looked at different types of data platforms characterizing over 300 melanomas. And what we found was that melanoma is made up of different subgroups, some of which are known to respond to certain therapies. For example, this BRAF subtype is the subtype that responds to therapies. Some of the other subgroups that we identified, it's not clear exactly what's happening. And we also identified a number of biomarkers that were associated with response. But one of the limitations of this study is we were studying treatment-naive cancers. And as I mentioned, there are a number of really great therapies into the clinic. And so for us to understand why certain patients are responding, why others aren't responding, to understand resistance mechanisms, and to develop new therapy, we really need to carry out our molecular profiling studies from patient samples that are being treated with these new exciting therapies. And this is one of the reasons why I came to Montreal area and the McGill community. When I interviewed here, there were a number of talented research scientists in oncology, dermatology, pathology, surgery that wanted to collaborate in this area. And many of these investigators are associated with the affiliated McGill University hospitals. And one of the main reasons why I set up shop at the Goodman Cancer Research Center, where there are so many investigators here with experience and authority in cancer biology. We have over 35 associate members, and when you count the life science complex, it's up to 70. And what we could do is take the information we were getting from patient samples and recreate these in cell lines to understand the biology behind the genetic alterations that are found in, that we are finding in our sequencing and profiling studies. We have expertise to develop new animal models, creating little avatars for patients to understand therapy response and outcome. And we do have strength here in genomics and bioinformatics, especially with the close collaborations with the Genome Center. But we've also developed a number of platforms and core facilities that are looking at different aspects of the tumor. And I won't have time to go into the details, but these include the immunophenotyping platform that we have at the Goodman Cancer Center and a metabolism core, which has been very successful and historically been a big part of the Goodman Cancer Research Center. Now, where this project really took off was through funding. So 2 summers ago, the scientific director of the Terry Fox Research Institute came to Montreal. They were interested in funding collaborative projects, as they already funded a collaborative project between BC and Ontario. And actually, one of their goals is to establish a consortium across the country to share molecular profiling data that we were generating, for example, with melanoma, but for other cancers as well, and to share it across all cancers across the country. And from this initial meeting, what formed was the Montreal Cancer Consortium, involving 4 major research hospitals, the Goodman Cancer Research Center, IRIC, and the Genome Center. And what our goal is to focus on projects to improve precision medicine, to establish a collaborative framework for profiling cancers, to specifically investigate mechanisms of drug resistance and response. And there's 4 demonstration projects that were funded, including a focus on melanoma, which was my interest, leukemia, and we are setting the foundation for future projects in other cancer. So just to give an idea of the power of the consortium, the 4 research hospitals involved, the CHUM, HMR, Jewish General Hospital, and MUHC, collectively see over 17,000 new cancer patients annually. That's one of the biggest group of patients found throughout Canada and would be competitive with institutions across the world. We have experience in clinical research, basic and translational research, especially experience in bioinformatics, as well as experience translating these findings with partnerships with industry. We already have co-sponsors with the Rossi Cancer Network, C3I, Exactis, and Avato, and co-funding coming from Oncopol and Genome Quebec. And so what we've done is brought our collective expertise in all different types of analysis of tumors. You cannot really do this in one lab, and so what we've done is brought cutting-edge technology and experience from various investigators at the Goodman Center, the Genome Innovation Center, the CHUM, HMR, Jewish General, MUHC, and IRIC. And we're all bringing our collective expertise to address the question, looking at melanoma patient cohorts being treated with immune therapy and targeted therapy, to understand what is happening at the molecular level, understanding what's happening at the patient level regarding their circulating immune system, to identify biomarkers for better patient selection of patients that are likely to respond or not respond, understand side effects of certain drugs related to the therapy, and identify new targets in order to advance the field and reach our goal of developing new strategies to improve outcome including long-term durable responses in the majority of the patients. And this is how we're realizing this goal. The Cancer Consortium pilot project has over 50 investigators currently involved in the project. And again, the goal is to set up a Montreal Cancer Consortium. There's already been funding for Ontario and BC, but the goal from this project is to link all cancer centers across the country. And so future projects are being funded in the Atlantic provinces, in the prairies, and again, we're working to share all this molecular data across centers in order to collectively advance outcomes for cancer patients. So with that, I'd like to thank my research lab. I've been very lucky to have a dedicated group and all the funding agencies. I'd like to thank the Goodman family and Dr. Bellini for setting up the infrastructure to make this possible, and the Goodman family for their continued support of our research program. So thank you.
Any questions for Dr. Watson? Perfect.
Thank you very much for including me in the celebrations of this beautiful building. The first time I saw the Bellini building was in 2010 when I was in Montreal for a different purpose. And I remember leaving the building and thinking, this is phenomenal. I wish one day I could work in an environment like this. So when McGill called a few years later and offered me a position in the Department of Biochemistry, it was obviously a no-brainer to join the department and to join the chemical and structural biology theme. So what I've summarized here is some achievements that have happened in the chemical and structural biology theme, and this is by no means complete, but I focus on research areas that investigators that are physically located in this building have accomplished. And these have been groundwork on many research areas that have resulted in seminal contributions to the literature, but also collaborations with other themes within the life sciences complex. We have enriched tremendously the research programs of all the investigators in this building. In my mind, even more important from these collaborations have stemmed our ability to bring state-of-the-art infrastructure to the building, secure training grants to train the next generation of scientists so that the wealth of information can keep progressing, and also the creation of the Center for Structural Biology, which was created a few years back and now includes over 30 investigators in 3 different faculties. There were 2 other things that were very attractive to me to join McGill University. One was, of course, the Goodman Cancer Research Center, because in my lab, we're interested in the mechanisms that repair DNA, and the possibility to interact with researchers in the Goodman Cancer Center was a big plus for me to join McGill. The second one was the Facility for Electron Microscopy Research, and in the last 10 years, electron microscopy has gone on its own revolution, something that colloquially is called is known as the resolution revolution. Less than 10 years ago, cryo-EM was able to yield structures that were only low resolution and provide limited mechanistic understanding of these proteins. But in the last 10 years, we've gone from this low resolution structure to pseudo-atomic resolution structures. I'm showing you here a picture of the Titan Cryos because this is one of the microscopes that has enabled this revolution. McGill had the vision to invest in this technique very early on and brought the first Titan Cryos microscope to Canada. And through the years, they've invested on building the microscopy field. They've recruited very heavily on that area. And now McGill has become one of the strongest hubs in North America for cryo-electron microscopy research. So in my lab, we're interested on the mechanisms that maintain genome integrity. So when we think about our DNA, we normally think about that duplex DNA that is extended and very nice and protected. But in reality, our DNA in all our cells is subject to constant insults, external and internal insults that will damage our DNA. Chemicals, radiation, oxidative agents. The very own process of copying the DNA might be mutagenic because we can introduce errors in our DNA. So each and every cell on each and every organism have developed mechanisms to detect immediately these problems and correct them promptly so that they don't become major issues on the cell. Another aspect why this is important is because most tumor cells have lost at least one of these repair activities. And while this often provides a proliferative advantage to these tumors, it's also an Achilles heel that we can use for therapeutic purposes. So, and this was very nicely exemplified by combined treatments using PARP inhibitors, which will block base excision repair, to treat tumor cells that were BRCA1 and BRCA2 deficient, which basically means that they cannot perform homologous recombination. However, to fully exploit the potential of these approaches, we need to understand how these pathways work, and we need to identify what are the best targets on each pathway that can be targeted for therapeutic purposes. So in the next few minutes, I'm just going to give you a couple of vignettes of work that we've done in the lab. The first one is on the mechanism of DNA, of correcting DNA mismatches. DNA mismatches are errors that have escaped polymerase proofreading. So polymerases are very good at copying our DNA, but every now and then we'll make a mistake. Those mistakes need to be promptly corrected because they are not a lesion per se. Both bases are perfectly fine, it's just that they don't pair. So if we don't correct them soon and the cell undergoes another cycle of replication, the two strands are going to meld and they are going to look perfectly fine, and they will become a mutation engraved on that DNA. So the DNA repair, the mismatch repair system identifies these lesions, fixes them, and in doing so, it increases replication fidelity and prevents genome instability. When we have inactivating mutations on DNA mismatch repair genes are the underlying cause of Lynch syndrome, which is characterized by a much higher predisposition of developing numerous cancers, primarily colorectal cancer, but also endometrial, stomach cancer, etc. So in my lab, on the last few years, we've done quite a bit of work at the step of the reaction in mismatch repair that recognizes which one is the new strand from which one is the template strand. And this is the most important step because those bases are fine, so you need to understand which one is the new one. So we've done quite a bit of work on understanding how these three proteins, mutase, mutel, and the beta sliding clamp, or PCNA, come together to identify which strand is the new one and introduce a nick. And that's the signal to correct that strand. So we were able to solve the structure of this endonuclease domain. We were able to discover the role of this protein in this reaction. And what I'm showing you here is when we put the two proteins together, what we were able to understand is that when the two proteins come together, there's this massive conformational change that presents the endonuclease site of mutel to the central cavity of the beta clamp. And that's the way that the beta clamp is able to present the newly synthesized strand to mutel. So the second vignette is something completely different and something completely new. And I wanted to present it here, although it's not quite ready for prime time, because it is this work that we've done at McGill University and that we wouldn't have been able to do elsewhere. So these are the mechanisms of something that we're interested in is how the TN7 transposon targets different sites on DNA. So most mobile elements have a core transposase and an adaptor protein. And what they do is they take the DNA from one place and they direct it to another place. And then these transposons were widely spread in bacteria, humans, wherever. They also carry a variable region that can piggyback other genes into different places. And in bacteria, normally this is genes that confer antibiotic resistance. So these are the core architecture, but TN7 has a more sophisticated targeting mechanism. Instead of using this adapter protein to target DNA, it has two additional proteins, TNSD and TNSE, that targets different sites, either a chromosomal attachment site with very high frequency or structures that are replicating DNA. So these are interesting because this system has been used to develop genetic tools. So a variant of this core machinery with a mutation on this adapter protein has been used in many labs. Most of us have used it to design, to perform random mutagenesis screens. And the TNSD pathway, it's normally used for cloning techniques and producing recombinant baculovirus so that we can do recombinant expression. However, these tools are not very efficient. And the reason that why we don't have better tools is because we don't understand really how these proteins target DNA. So one of the goals in the lab is to understand how these proteins target DNA and develop better tools that are more specific and more efficient. So we started working on TNSE, and I'm not going to show you this data, but I'm just going to finish with this structure. This is the structure of a variant of this mutant protein that can bypass the need for these targeting proteins. This is data collecting in the Titan Cryos here in the Strathcona building at 4 angstroms resolution. This is the structure of TNSC bound to DNA. So this is allowing us to understand the mechanism of how this protein targets DNA. And I've been told that this is the first ab initio and de novo protein that has been determined at femur. So we're very proud that we were able to contribute to this. And with this, I'm going to stop here. I'm going to thank my lab, similar view to the one that David used, but with all graduate students and undergrads in the lab, and also funding for supporting our research program. Thank you very much.
Do we have any questions?
It's a great honor to be here and represent my research theme and discuss with you some of my personal research work. In hockey and in basketball, we are talking about MVPs, most valuable player. And I personally think what's the MVP here is our facilities and our excellent enabling technologies that enables every researcher in this building to be a star in the game. So thank you very much for everyone who contributed to this excellent facility, especially our generous donor. So when I started my research training back in Vienna, I was interested how is our body actually able to mount a successful immune response that we can fend off viruses and bacteria. And what we know is that our body recognizes these microbes by very specific protein families, such as toll-like receptors, or inside of the cell, such as cytosolic node-like receptors, and then instruct signal that we can create immunological memory, a T-cell response, an antibody response, that we then, and this information we can then, utilize to create better vaccines. And when I was a master and PhD student, I worked in Vienna at a biotech company called Intercell, which is now bought up and renamed as Valneva, and we developed a novel vaccine adjuvant that is now in phase 2 and phase 3 clinical trials for tuberculosis and other infectious diseases, and utilized 4 patents. And then I was really interested in working out the downstream mechanisms that are required to instruct this immune response, and went on to do training at the Institut Pasteur and the University of Toronto. And I'm thankfully, that was quite fruitful and attracted interest in McGill of bringing me here. So what to the complex traits group located here on the 3rd floor of the Bellini. So the group is now consisting of 35 members. We're very interdisciplinary. Members are in all 6 departments: microbiology, immunology, human genetics, medicine, biochemistry, physiology, and computer science. And we collaborate nationally as well as internationally with key players in our field. So what really kind of attracted me to come here and work in this group of excellent scientists is the conceptual framework that was put forward. We're looking at the phenotype of a disease, for example, malaria, looking at the genetic background, what is known from animal models, what is known from human genome-wide association studies, and then introduce gene defects in mice and study the phenotype in these disease models to actually find the cellular and molecular mechanisms that is underlying this defect, and then come up with novel strategies and therapies to develop new drugs, hopefully successful in human therapy. So when I interviewed here, that really attracted me because, this wealth of information and the possible models to work in infectious and inflammatory disease biology was excellent for me as a cellular and molecular immunologist. So the group has gathered a lot of technologies and resources, either in mouse genetics, several bacterial, viral, parasitic, helminth, and fungal infectious disease models in the mice, as well as more and more importantly also chronic inflammatory diseases such as inflammatory bowel disease, multiple sclerosis, or asthma. So personally, I'm really interested in a type 2 immune response and its associated pathologies. So a type 2 immune response has evolved over time to restore damaged tissues, broadly speaking. And in parallel, it's utilized to instruct when people are infected with helminths, with worms, to clear those. But when the type 2 immune response is deregulated, we are facing severe developmental disorders such as allergy, asthma, atopic dermatitis. But we also know now more and more that type 2 immunity is really important for the browning of our fat, meaning that metabolic disorders such as obesity actually play an important role when we study type 2 immunity. We also know that the type 2 immune response predisposes developing cancer to create a pro-cancerous niche. So studying the mechanisms might be beneficial when we come up with combination therapies in cancer. But also type 2 immunity, as I said, is really important for tissue restoration. We think that the molecular and cellular players in this type 2 immune response is really important when we consider neuroregeneration of damaged neurological tissues. So what was known in type 2 immunity that, upon tissue damage or infection or allergen trigger, that at the epithelium there are certain molecular mechanisms triggered that lead to a cytokine release that then instruct a T cell response and a B cell response, possibly very well known in the communities that type 2 immunity leads to immunoglobulin E, which really makes us, are the signs of allergy. But what was, when I started here, there was a new cell type discovered, which is referred to as the group 2 innate lymphoid cells, which produces very potently and very fast a set of cytokines that instructs this type 2 immune response. And my lab over the last years has generated cell culture models of mouse and as human ILC2s, and we can utilize that now to do drug screens, to study the cellular and molecular mechanisms, how to target very early on this cell type that we can dampen all these associated pathologies. So what has been built up here and what I think this facility has really enabled to bring together technologies in cellular and molecular work, regardless of every theme, that enables the whole community to have state-of-the-art access to these technologies. In our group, we're mostly utilizing, of course, imaging, such as classical microscopes as well as in vivo microscopes. And we've built up, in-depth cell phenotyping cells from infected tissues, from, patient samples, that we can study the different phenotypes and associate them with disease outcomes. And of course, study, inside of the cell what's actually going on and link all that information back to the genetic background of the disease. So what I personally have contributed here, classical flow cytometry, which is the technology that studies, single cells and phenotype them in detail. We've brought here on the third floor all, flow cytometers, the old and the new generation together, and we, created a platform that serves now about 70 different laboratories and 250 users. So this facility has enabled that. Where do we want to take that to the next level? What I really think, thankfully, this year's Nobel Prize in Medicine with cancer immunotherapy has brought the term immunotherapy to the broad public. And I think this is very important because immunotherapy is not only important in cancer immunobiology, but in immunology and in disease biology as well. 10 years ago, we did not think that, disorders such as obesity has a very important immunological aspect. Now we know that. And I think that studying the immune angle in every disease is important. So what we have not attempted well in the clinical picture is what is actually happening at the cellular and molecular level in depth when we treat people with certain drugs that have success, but they have limited success. So I think what we really need to learn is to do deep molecular and cellular phenotyping of people when they get certain drug treatments and link that with the genetic information of the pool. So what we are trying to put forward is a drug and discovery development pipeline where we are applying, for a CFI and we hope that the faculty in Central McGill will look favorably at that. And the most, like the two technologies that I think will bring us to this next level is an in-depth tissue analysis system as well as single-cell genomics. So single-cell genomics will enable us that we actually look at single cells from a deceased patient at different before and after drug treatment, and we then can not only quantitatively look at the different single cells here, but we can also look at them, what other genes do they express, and we can link the expression of the gene pattern at the single cell level to the disease and to the drug treatment. In parallel, we want to look at how do these cell types then actually localized in the diseased tissue. And there are newer tissue analysis systems in place where we can do that. And we think that the combination of these new technologies will enable us to bring disease research to the next level. With that, I have to thank my group, which enabled me to come here to McGill and enjoy this great facility and this great interactive environment. Thank you very much.
Thank you very much to the Bellini family, the Goodman family, and also the representative of the government, and to all our supporters and friends. We will have a special tour of the complex for you this afternoon. For all our guests, please enjoy coffee and the poster session that will be in this room and throughout the complex until lunchtime, which is at noon, when you can make your way back here. The exact location of the poster session is on the screen, and this afternoon's presentation are in the Howard Palmer Amphitheater and features Michel Tremblay and Claude Gingras from Lunafeld Tannenbaum Research Institute, Mount Sinai Hospital, one of our own, as well as several students. The order of proceeding is on the screen as well, and our end of celebration cocktail reception will take place at 4:30 today. Merci à tous et passez une excellente journée.